In Chapter 7, we're going to cover cellular respiration and fermentation, which is basically how our cells do work. Cellular respiration is what our cells do in order to make energy so that our body can do the things that it needs to do. And this subject, at least for myself, was always a little difficult for me to understand when I was taking introductory biology courses. So hopefully we can keep it as simple as possible so that it's the easiest for you to understand. We're not going to go super in depth of the parts of cellular respiration because that's something that you will cover if you take organic chemistry. But instead, we're going to just really try to keep things very generalized for you. So, as you know, all living cells require energy from something outside of itself. Cells can't make their own energy necessarily. So, we have to eat things, that food gets broken down, and it gets turned into chemical energy that our cells can use. The general flow of energy in an ecosystem is that it starts as sunlight and it leaves as heat. So when you're moving and doing work, you are generating heat. When something dies, it's cold to the touch because energy is not being exerted, work is not being done. And that's one reason why they get cold. Photosynthesis is what we'll learn about in Chapter 8. It generates oxygen and organic molecules, which then we consume to use for cellular respiration. So photosynthesis and cellular respiration absolutely go hand in hand. Cells use chemical energy stored in organic molecules to regenerate ATP. ATP is chemical energy for our cells.
Could a carbon atom that was once part way, carbon that was in the air is now part of the plant.
So this is the basic overall figure that illustrates how energy enters a system. So we have light energy that comes in through the sun. Plants have chloroplasts, which take in that light energy and convert it into some sort of an organic molecule, also giving off oxygen at the same time. That oxygen and those organic molecules are utilized by the workhorses in our cell called the mitochondria, and they go through something called cellular respiration. Two byproducts of cellular respiration are carbon dioxide and water, which is used by plants in photosynthesis. So this is a very cyclical cycle right here. And then another byproduct is ATP, and that's what our cells use to do work. So I would know the byproducts in general of both of photosynthesis and cellular respiration, the end products here. I would know that light energy is the ultimate source of energy at the start of every ecosystem, and that all energy is given off as heat, taken in as light and given off as heat. Catabolic pathways involving electron transfer are essential processes in cellular respiration. So if you remember, catabolic pathways give off heat. They don't take in heat, which would make sense. If heat is one of the end products, then we've got to have a catabolic pathway. The breakdown of organic molecules is exergonic. Fermentation is the partial degradation of sugars that occur without oxygen. And so we'll talk about that a little bit later. Aerobic respiration consumes organic molecules and oxygen and yields ATP. Aerobic respiration is what most of our cells ultimately do. Anaerobic respiration is similar to aerobic respiration, but it consumes compounds other than oxygen. Anaerobic respiration might be something that happens when you're working out really hard and you've used up all the oxygen that's available to your cells. And one of the byproducts of anaerobic respiration is lactic acid, which is what burns in your muscles. So you know that you have gone through anaerobic respiration if you feel sore muscles. Cellular respiration includes both aerobic and anaerobic processes, but is often used to refer to aerobic respiration only. Although carbs, fats, and proteins are all consumed as fuel, it's helpful to trace cellular respiration with the sugar glucose because it's the simplest form of energy that our body takes in. All other foods, carbs, fats, and proteins, eventually get broken down into that simple element of glucose. But when we look at cellular respiration, let's look at it very simply. And that would be one reason also why more complex carbs, fats, and proteins are going to give you longer term energy than very simple sugars, like just having a candy bar, because it gets broken down really quick and the energy is gone. Carbs that are more complex, fats and proteins, are going to take longer to get broken down, so it's almost like a longer release form of energy. Now, I don't know if we've talked previously before or not about redox reactions, but the transfer of electrons during chemical reactions releases energy stored in organic molecules, and this released energy is ultimately used to synthesize ATP. So it's an oxidative and reduction or redox reaction when we go through cellular respiration. If you remember, chemical reactions that transfer electrons between reactants are called oxidative and reduction reactions or redox reactions. So you're moving electrons back and forth. And if you're being oxidized, if it's an oxidation, then you've given up an electron. If it's reduced or it's a reduction, then you have gained an electron, which means that you now have a more negative charge, which is why you get that term reduction. So reduced means you gained, but you have to think about what you gained. You gained an electron, which means your charge is lower. So in this example, this is a redox reaction, right, where sodium has given up, it has lost an electron, so it's become oxidized, and chlorine has been reduced, it has gained an electron. This is the reduction part of the reaction. Somebody gets an electron, somebody loses an electron. If you get it, you've been reduced. If you've lost it, you've been oxidized. So the electron donor is called the reducing agent, and the electron acceptor is called the oxidizing agent. So although chlorine is reduced, it's the oxidizing agent. And although sodium has been oxidized, it is the reducing agent. The electron acceptor is called the oxidizing agent. Some redox reactions do not transfer electrons but change the electron sharing in covalent bonds. And one example of that is between methane and oxygen. Not really that important right here. But here you can kind of see what it looks like. It's really just moving around these electrons as opposed to giving one up. So redox reactions that move electrons closer to electronegative atoms like oxygen release chemical energy that can be put to work. So ultimately, the important part of understanding a redox reaction is that chemical energy is released during this transfer. So during cellular respiration, fuel, like glucose, is oxidized and oxygen is reduced. I would definitely remember that fuel, glucose, is oxidized, oxygen is reduced. Oxygen takes on an extra electron, glucose gives it up. Organic molecules with an abundance of hydrogen, like carbs and fats, are excellent fuels because there's all those hydrogen ions that they can be moved around and given up, right? As hydrogen is transferred from or to oxygen, energy is released, and that can be used in ATP synthesis. This is looking at the very, very basics of cellular respiration. When we start looking really in depth at the images of them, you're going to understand what this means quite a bit more. So again, to rephrase it using the chemical reaction, glucose gives up hydrogen. So it gives up the electrons, it becomes oxidized. Oxygen becomes reduced. It gains hydrogens, it gains electrons. And when this occurs, energy is also made. In cellular respiration, glucose and other organic molecules are broken down in a series of steps. And we're going to talk about all of these steps. And I want you to know what goes into each step and what comes out of each step. But I don't really care that you know every single piece of the steps within those. Electrons from organic compounds are usually first transferred to NAD, which is a coenzyme. And as an electron acceptor, NAD functions as an oxidizing agent during cellular respiration because it will turn into NADH. It has been reduced because it has gained an electron in the form of that hydrogen, right? Enzymes called dehydrogenases facilitate the transfer of 2 electrons and 1 hydrogen ion to NAD, and then 1 hydrogen ion is released in this process. So this is really just the chemical reaction, what NAD looks like, this little, what's closed up in this box, and then what it looks like once it's become reduced and gain those hydrogens. So NADH passes electrons to this thing called the electron transport chain. We're going to talk about that. It's like the last part of cellular respiration. And as the electrons are passed to increasingly electronegative carrier molecules down a chain, it causes this series of redox reactions, and tons of ATP is given off. So as the electrons move down the chain, it ends up cranking this little pump, and then you release a ton of ATP. So in cellular respiration, this is a very generic way to look at it, but we have these hydrogen ions that bounce down an electron transport chain and as a result, they're releasing tons of energy in the form of ATP that now our body will use to do stuff. Harvesting energy from glucose has 3 stages. We have glycolysis, the citric acid cycle, and then oxidative phosphorylation. And that's where you've got the electron transport chain that we alluded to a little bit earlier. So glycolysis breaks down glucose into 2 molecules. Those 2 molecules then go through the citric acid cycle and they get broken down even further. And then oxidative phosphorylation is where all the electrons start rolling down. So once again, there are 3 parts to cellular respiration. Glycolysis is first, then there's the citric acid cycle, and then there's the electron transport chain or oxidative phosphorylation. And throughout the book, they're going to be color-coded blue and orange and purple for you. So glycolysis occurs outside of the mitochondria. And basically, it takes glucose, splits it in half, and turns it into pyruvate, 2 pyruvate molecules, and makes a little tiny bit of ATP, but not enough to really do very much. Its purpose is to get to this next step so that you can get to another step to get to a final step to make a ton of ATP. So once you pass through the mitochondria, then you have the citric acid cycle, which is inside the mitochondrial matrix. And then finally, we have oxidative phosphorylation, which goes across the membrane of the mitochondria, the inner membrane of the mitochondria. And this is where you have tons and tons of ATP. We call it oxidative ATP. There's substrate level ATP, which is in glycolysis, and the citric acid cycle. Oxidative phosphorylation accounts for 90% of the ATP generated by cellular respiration. So it is the most important, but we would never get to that step if you didn't have the 2 previous. A smaller amount of ATP is formed in glycolysis and the citric acid cycle. Know that we call that substrate level phosphorylation. In this process, an enzyme transfers a phosphate group directly from a substrate molecule to ADP. So it turns ADP into ATP, right? For each molecule of glucose degraded to CO2 and water by respiration, the cell makes up to 32 molecules of ATP. So 32 is an important number to remember. How much ATP is made from glucose? 32. Glycolysis literally means sugar splitting, and that's what it does, right? It turns glucose into 2 molecules of pyruvate. So what I would know for glycolysis is what goes in, what comes out, and what is made. Glucose goes in, 2 molecules of pyruvate come out, and 2 ATP and 2 NADP are made. So glycolysis occurs in the cytoplasm. It occurs outside of the mitochondria. I would also know what goes in, what comes out, what is made, and where it happens to. So it occurs in the cytoplasm outside the mitochondria, but inside the cell. And there's 2 parts to it. We call it the energy investment phase and the energy payoff phase. Just like anything, even in business, you have to spend a little bit of money before you actually start to make money, right? You have to buy materials. You have to pay rent for a building. There's money that has to go in to be invested into something before you get a payoff phase. So the net energy yield is 2 ATP plus 2 NADH per glucose molecule. And that's because 2 ATP went in, 4 came out, but your net is 2, right? Glycolysis is going to occur whether oxygen is present or not. So that's an important thing to remember. And then, so here's glycolysis again. It's occurring outside of the mitochondria. So our energy investment phase, we have to use 2 ATP to get things rolling. And then there's the payoff phase. We've made ATP and we've made NADH. So know what the net is, know what goes in, which is glucose, know what comes out, 2 pyruvate plus water. Know that we make 4 ATP and 2, but we use 2 ATP, so our net is 2. And then with NAD, we've made 2 NADHs, which will be important later on because those hydrogens can be used. So if you look at it, ultimately what I really want you to look at is that glucose comes in. I don't care about each individual step here. That's something that you're going to cover in organic chemistry. So really, ignore that, ignore that, ignore that. Just know that in comes glucose, a little bit of ATP is added, and then our end product is we have 2 pyruvates, and we also have we have ATP that's formed also, ATP and NADH. So when oxygen is present, pyruvate will enter the mitochondria, and then that's where oxidation of glucose is finally completed. So there's a tiny little step before citric acid cycle begins. Pyruvate has to be converted into acetyl-CoA, which links the glycolysis and the citric acid cycle together. So a really good trick question would be, true or false, the end product of glycolysis is what goes into the citric acid cycle, and that's not actually accurate because that pyruvate has to be converted into acetyl-CoA first. So what enters the citric acid cycle? Acetyl-CoA. So pyruvate, and this happens across the membrane. So pyruvate moves across, it turns into acetyl-CoA, and then now we're inside of the cytosol of the mitochondria. We're in the citric acid cycle inside the mitochondria, and we just have this cycle. Acetyl-CoA is just circling and circling around, continuing to move and move, and in the process we're kicking off a bunch of NADHs, even something called FADH2. But what's really important in these is the fact that we have hydrogen molecules. Those are what are significant about these letters that we're spitting out. ATP, a small amount of ATP is also made, and then this is where we give off our CO2. CO2 is given off during the citric acid cycle. So pyruvate comes in from glycolysis. Pyruvate oxidation happens so that we can enter the citric acid cycle. You might also see it called the Krebs cycle. And so again, CO2 is produced, and that's what's important. What we've generated ultimately is 1 ATP, 3 NADHs, and 1 FADH2 per turn. So, I mean, from one glucose molecule in the citric acid cycle, how many ATP do you make? Well, it's kind of a trick question, right? Because it will pump 2 times, won't it? So make sure that if there is one of those kind of tricky questions on the quiz or on the exam, you read it carefully and find out, is it asking about glucose, or is it asking about the acetyl-CoA or pyruvate? There's going to be different answers. Citric acid cycle has 8 steps. I don't care that you know each one of those 8 steps. Again, you will learn that kind of stuff when you get into organic chemistry. Just know that it has a bunch of steps in there. And this is what they look like. When you get into organic chemistry, you have to know each of those individual steps and what's happening in each of those steps. And then there's just a bunch of redox reactions also that are happening during the citric acid cycle, converting NAD to NADH, and then the FAD to FADH2. So during oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. So NADH and FADH2 are ultimately what make most of the energy that's extracted from food. And it's because they're giving off those hydrogens that they have accepted. So they're called electron carriers, and they donate their electrons in the form of hydrogen to the electron transport chain, which powers ATP synthesis. The electron chain is located in the inner membrane, or the cristae, of the mitochondria. And so the chain's components are a bunch of proteins, basically. And they alternate, they reduce and oxidize different states as they accept and donate the electrons. The electrons drop in free energy as they go down the chain, and then they're finally passed on to oxygen, and they eventually will form H2O, because remember, the electron is in the form of hydrogen. So electrons are transferred from NADH or FADH to the electron transport chain, and then the electrons are passed through a number of proteins, including cytochromes, eventually to oxygen. So the electron transport chain generates no ATP directly, but it breaks the large free energy drop from food to oxygen into smaller steps that release energy into manageable amounts. So oxidative phosphorylation occurs in the inner membrane of the mitochondria. So it's still inside the mitochondria, but it's going across that inner membrane. And here's the free energy that's being released, or the energy that gets released, ultimately making water down here. And so NADH becomes oxidized, or I'm sorry, yeah, it becomes oxidized, and it gives up its hydrogen, and those hydrogens go rolling down this electron transport chain through all these little proteins. And then eventually, right, the electrons spit out, it binds with oxygen, and then it finally makes water. The electron transfer in the electron transport chain causes the proteins in the pump, those little globular purple things to pump hydrogen from the matrix into the inner membrane space. Now, if we go back, the inner membrane space would be, I guess this is probably the best picture. So we've got our, we have our outer membrane here and our inner membrane right here. And so this across here, all this is that inner membrane space. And then hydrogen moves back across the membrane. It goes through this thing called the ATP synthase, and a bunch of ATP is made. Synthesizing means to make something. And so as the hydrogens get kind of shoved through this ATP synthase here, it forces this sucker to turn, and it makes tons of ADP be converted into ATP. The energy stored in a hydrogen gradient across the membrane couples the redox reactions of the electron transport chain. Basically, well, there's a video that's going to make a lot more sense than just trying to read these words and explain it. But here we have NADH and FADH2 that are being oxidized. They're giving up their hydrogens, right, to the chain. It's pumping the hydrogen, so you have this space over here with all these hydrogens, and it doesn't like that because it's higher in hydrogen here than it is down here. And so as a result, these hydrogens kind of gather all over here, and then they get shuttled down this ATP synthase, which causes that to turn and make a ton of ATP. So NADH and FADH2 giving up their hydrogens form an imbalance of hydrogen on one side of the membrane. Those hydrogens want to get back across, and when they do cross through the ATP synthase, that thing turns and twists and produces ATP. During cellular respiration, most energy flows in the following sequence: glucose, NADH, electron transport chain, proton motive force, and then ATP. So that proton motive force, of course, being the synthase here. About 34% of the energy in glucose molecules transferred to ATP during cellular respiration, and it makes about 32 ATP molecules for us. And there's several reasons why it changes a little bit. We're not even going to attempt to get into that because that's not even necessarily known and way beyond what is what we need to know in an introductory biology. So here you go. Here's a really good overall picture of what cellular respiration looks like. Glucose comes in, converts to 2-pyruvates. This happens outside of the mitochondria. We have made 2 ATP and we've made 2 NADHs. We did have to take in some ATP, right? Then those 2-pyruvate are converted into 2-acetyl-CoAs because that is what has to be accepted by the citric acid cycle. That citric acid cycle pumps, makes a little bit of ATP, but what it makes that is most significant to us is that it has a bunch of NADHs and FADH2s. Those hydrogens in the NADH and FADH2 get taken off, oxidative phosphorylation happens, all those hydrogens accumulate on one side of the membrane, and then they want to come across, so they get pumped through, and as they get pumped through, it turns that electron transport chain, or it turns that synthesis, and it spits out a ton of ATP molecules for us. Most cellular respiration does require oxygen to produce ATP, but sometimes we don't have oxygen available to us. So without oxygen, the electron transport chain won't operate. In that case, we end up having fermentation or some sort of anaerobic respiration that is used to produce ATP. Anaerobic respiration uses an electron transport chain with the final electron acceptor that's not oxygen. And fermentation just uses substrate level phosphorylation instead of an electron transport chain to generate ATP. So know the difference between the two of them. Anaerobic respiration still uses an electron transport chain, but it doesn't use oxygen as that final acceptor molecule to make water. Fermentation uses substrate level phosphorylation, so it only does glycolysis, or what's the other one? The other one was citric acid cycle. So fermentation consists of glycolysis plus reactions that regenerate NAD, which can be reused by glycolysis. So there's alcohol fermentation and then there's lactic acid fermentation, both of which humans use to our benefit, right? Alcoholic fermentation we use to make wine and beer and alcohol. And then lactic acid fermentation our body uses when we are low on oxygen, working out really hard. There's not available oxygen for our body to take in. And that's when you feel muscle soreness because it's actually lactic acid tears in your muscle and lactic acid that builds up in those muscles that causes that kind of burning sore feeling that you feel when you have worked out. In alcohol fermentation, pyruvate is converted to ethanol, and ethanol is, of course, our alcohol. So it happens in 2 steps. The first step, you release CO2 from pyruvate, which would be the bubbles, which would be the carbonation that happens in the beer. And the second step reduces what's formed into ethanol, makes the ethanol. So alcohol fermentation by yeast is used in brewing, winemaking, and also baking. Because of the CO2 that's released for baking, it helps bread rise because air is being released into the dough. Okay, so this is the overall of alcoholic fermentation. Glucose comes in, glycolysis occurs, pyruvate gets turned into acetaldehyde, and then that is made into ethanol. So your end product is ethanol, some energy, and lots of NADHs that can be used back again in glycolysis. But in lactic acid fermentation, pyruvate is reduced by NADH. It forms lactate as the end product. We don't release any CO2. So lactic acid fermentation is used by some fungus and bacteria, and that's actually how we make cheese and yogurt. But our muscle cells also use it when oxygen isn't readily available to us. So know the difference between the two and understand that the two are utilized when and occur when oxygen is not present. All use glycolysis to oxidize glucose and other organic fuels to pyruvate. So we're comparing anaerobic and aerobic respiration. So glycolysis is always going to occur. NAD is the oxidizing agent in all 3 as well, is always going to occur. But the mechanism of NADH oxidation is going to differ. So in cellular respiration, transfers the electrons from NADH to the carrier molecule to the electron transport chain. But in fermentation, the final electron acceptor is either pyruvate or acetaldehyde because you don't have the electron transport chain when you have aerobic respiration occurring. Cellular respiration produces 32 ATP per glucose molecule, but fermentation only produces 2. So it's really not effective at all. It's much more efficient to utilize oxygen to make energy. Long term, you could not sustain yourself without oxygen. We also have a couple things called obligate anaerobes and facultative anaerobes. Obligate anaerobes carry out only fermentation, and they can't survive in the presence of oxygen. They are obligated to do anaerobic respiration. But yeast and bacteria are facultative anaerobes, which means that they can do either or. And if they're facultative, then pyruvate is a fork in the metabolic road that leads to the 2 alternative routes. So you either use without or you use with. So facultative means you can facilitate whether you do use oxygen or not. Glycolysis is the most common metabolic pathway among organisms on Earth, which means that it probably evolved very, very early on in history. And early prokaryotes may have generated ATP exclusively during glycolysis because there wasn't a whole lot of oxygen in the atmosphere. And then also the location of glycolysis being in the cytosol instead of inside the mitochondria also indicates ancient origins because mitochondria didn't evolve until much later in eukaryotic cells than in prokaryotic cells. And then glycolysis and the citric acid cycle are major intersections to various catabolic and anabolic pathways. I would just understand that other stuff can arise from it. They're major intersections, but as far as really understanding it, just basically understand this picture gives you good overall, right? So if it's a simple sugar, it goes straight into glycolysis, right? But if you've got fatty acids or you have proteins, they come in and they come in a little bit later. And there's a lot of steps that happen here that we're not even looking at. So these proteins and these fats can be inserted into glycolysis and the citric acid cycle to make oxidative phosphorylation happen. But carbs are the simplest form and they just shoot right inside there, which is why they generally give you the shortest amount of